In this video, we will explore the functions of various bacterial cell structures, which are identified in the diagram and described here. We will discuss the structure and function of the capsule, cell wall, cell membrane, cytoplasm, ribosomes, plasmids, pili, flagella, and nucleoid circular DNA. Bacterial cells, also known as prokaryotes, are tiny unicellular organisms, typically not larger than 0.1 mm in length, making them about 1,000 times smaller than plant cells, and visible only under high magnification microscopes. Their cell walls are composed of peptidoglycan, a complex mix of proteins, carbohydrates, and lipids, and they may or may not be surrounded by a gelatin capsule. Inside the cell wall lies the cytoplasm, which can contain granules of glycogen, lipids, and other food reserves. The cell contains a coiled loop of DNA, but no nucleus, and is filled with ribosomes, which are smaller than those in animal or plant cells. Some bacteria have one or more flagella for movement and occasionally possess plasmids. The functions of bacterial cell components are crucial for the cell's survival and operation. The capsule serves as a protective layer, while the cell wall prevents the cell from bursting and allows water and salts to pass through its freely permeable membrane. The cell membrane controls what enters and exits the cell, protecting its contents. The cytoplasm holds all cell organelles and facilitates their functions. Circular DNA governs cell division, development, and overall activities. Plasmids carry genetic information essential for the bacterium's survival and reproduction. Ribosomes are responsible for protein synthesis within the cell, and flagella enable motility, allowing the cell to move through its environment, with structural and movement differences between prokaryotic and eukaryotic cells. Moving on to the division of a bacterial cell. Binary fission is the primary method of asexual reproduction in bacteria, beginning with a single bacterial cell. The process starts when the cell's circular chromosome replicates, producing two identical copies. The cell then divides into two daughter cells, each receiving one copy of the replicated chromosome. This cycle repeats, causing exponential growth of the bacterial population. As an asexual process, binary fission does not involve the fusion of gametes, and it occurs rapidly, enabling quick reproduction under favorable conditions. The daughter cells are genetically identical to the parent cell, with occasional mutations. Notably, the bacterial chromosome is not enclosed in a nucleus, a key feature of prokaryotic cells, and is coiled to occupy a small portion of the cell. Whereas, plasmids are small, circular DNA molecules found in bacteria, separate from chromosomal DNA, and capable of replicating independently. They are widely used in biotechnology for genetic modification because they can easily carry and transfer foreign genes. Scientists insert specific genes into plasmids and introduce them into host organisms, like bacteria, to produce proteins or other products. Plasmids play a crucial role in gene cloning, gene therapy, and recombinant protein production, such as insulin and vaccines. Their ability to manipulate and transfer genetic material makes them essential tools in biotechnology. The diagram shows how scientists insert genes into plasmids, enabling organisms to produce specific proteins or display desired traits. The gene is introduced into a bacterial plasmid, and as the bacteria multiply, they produce the desired products.